part of the transcribed RNA, there is also a non-conserved region. And this is what we hypothesize to be the spacer, the guide sequence that directs IACB to specific positions in the genome. And that turned out to be exactly the case. And so we test IACB and we show that IACB is exactly functioning like a RNA-guided Cas9. And so we then solved the cryo-EM structure for IACB. And you can see based on the structure of IACB and comparing that with Cas9, IACB is very small in the protein component. So these yellow parts are the protein. Cas9 is much larger, and the red domain that is missing IACB is the orange yellow domain that's on top over here. What is very interesting is that this red domain protein part in Cas9 is replaced by non-coding RNA domains in IACB. And so what really happened is that IACB, the ancestor of Cas9, was a small protein working with a very large non-coding RNA to form this ribonucleic protein complex. As evolution took its course, a lot of the non-coding RNA got replaced by protein domains to make it into a more robust RNA-guided nuclease that bacteria can use as a part of the immune system. So this sort of led us to understand the evolutionary origin of Cas9 a lot better. And the compactness of IACB potentially has useful applications for in vivo delivery because the viral vectors or the nanoparticles that are used to deliver these things usually work better when things are smaller. So this is one potential direction to further develop IACB. In addition to IACB, we also found the ancestral protein for Cas12, and that's a protein named the TMPB. TMPB is really interesting. It's probably the most abundant protein in all living organisms. In a single bacterial genome, you can find sometimes 500 copies of TMPB. So they are as abundant, if not more, than ribosomal genes. And so CRISPR Cas12 really came from this really broadly occurring protein. Another interesting thing that we reported recently was that there are CRISPR-like systems, TMPB derived, that are found in eukaryotes. Sometimes you can find them in single cellular eukaryotes, and then you can also find them in some cases, more rare, multicellular animals. So this plant here has these RNA-guided systems inside. And these systems are called Fenzor, and if you look at the cryo-EM structure for Fenzor, they look very much like the Cas12 proteins, and also very much like the TMPB. They have a protein domain with a large red domain, as well as a large non-coding RNA that we call omega RNA, to be able to direct this and to recognize and also cleave specific DNA sequences. So Cas9, Cas12, Cas13, IACB, TMPB, Fenzor, PNMA2, etc. They're all naturally occurring systems that provide very important functions in their native context. And what this really highlights is that if we further explore nature's diversity, there will likely be many more interesting systems that can be co-opted and engineered to do something that's useful to improve human health or improve other biotechnological applications. And so we're very excited to continue to explore that. This plot here shows the really rapid exponential growth in the amount of biodiversity genomic information that's being collected. And I think it's really the exploration of this that we're going to find a lot of the powerful tools that nature has developed that are encoded in these DNA double helices. So that's all for today. I just want to thank you again for the opportunity to share our work with you